So let's take a deeper look into the process of photosynthesis, that being the light-dependent reaction and the Calvin cycle. And then we're also going to look into a process called photorespiration and how plants kind of deal with that problem, which will lead us into C3, C4, and CAM plants. So when we talk about the light-dependent reaction, or the light reaction, this is happening on the thylakoid membrane, and it is taking place on the opposite sides of this membrane are the two substances, the lumen, which is on the inside of the thylakoid, and the stroma, which is on the outside of the thylakoid. Essentially what's happening is that we all know kind of have like the basis of how like a hydropower dam works, or just how a dam works. There is a border or a wall that is built, and we build up a bunch of water on one side, and then at certain points that water will flow through to the opposite side, and as it does, it turns a turbine and generates water. The same processes, essentially the same process happens here in order to generate ATP. And so as we go through that, it's a pretty good analogy as we go through this to realize kind of like there is a flow and a direction, just like upstream to downstream for or water in a dam. There's also going to be upstream and a downstream, or a high area concentration and a low area concentration within the lumen that's going to flow out into the stroma. And the border in between is the thylakoid membrane. And inside of this membrane, there's different proteins that are lodged and other enzymes and things that are going to be part of the machinery that we're going to use to produce the products of the light-dependent reaction. So remember, the parts of the light-dependent reaction was a little bit of ATP. We also got NADPH. We took in water, and we took in and released oxygen from this point, and also took in light. So you'll notice that two of these proteins are labeled. One's called PS1 and the other one's called PS2. So this stands for photosystem. And there are two of them. There's the PS1 and the PS2. We're going to start with the PS2. Why don't they call it PS1 if that's where we start? Well, it's because this one was discovered first. PS1 was discovered first, but it's the second step in the process. PS2 is discovered later on, but it's the first step in the process. So first thing that's got to happen is we got to have light coming in, right? Light-dependent reaction depends on light. And remember what we said, it's going to absorb this photon of light. That's the water coming up from the roots. When the water comes up from the roots, we said that the oxygen is not used. The hydrogen does the absorbing, so the oxygen leaves. That's a byproduct. That's plant parts. It's going to go back out and fill up the atmosphere. But the hydrogen that's left behind, kind of two things are going to happen. It splits into a proton and an electron. The protons are going to do their own thing, which we'll come back through look at later. But the electron, when it splits off, it's actually going to absorb that photon from the sun. And when it does, it becomes highly charged. So this is our highly charged electron. So the first thing that happens is this electron goes through a series of reactions. You hear me talk about the ETC later. There's a big one at the end of cell respiration, but any chain reaction involving electrons is electron transport chain. So it goes through a small little ETC, electron transport chain, and as it does, it's going to expend its energy. And what is it expend its energy doing? As it goes down, it pumps hydrogen ions, protons, from the stoma into the lumen. It just builds them over. It's building up a high concentration of hydrogen ions in the lumen. And so every time it goes through, it pulls one in, and then that electron ends up in photosystem 2. And photosystem 2 is a photosystem, so we're going to have a photon come in, right? Photon comes in of light. And it's going to recharge that electron and give it another burst of energy. But during this process, or the result of this process, it doesn't expend any of the energy. The electron actually will move up where it joins forces with two things, NADP and a hydrogen ion. When you take the electron and put it with that hydrogen ion, a proton, you make hydrogen. That hydrogen will then attach to the NADP to form NADPH. So all these things working together produce the NADPH that we get as the result of the light-dependent reaction. And this NADPH leaves this area from the thylakoid membrane and goes off into the stroma, which is where we'll have the Calvin cycle. So that takes care of at least the NADPH. We haven't made ATP yet, though. So let's go back at those hydrogen ions that we're building up in the lumen. So as they build up and build up, they get more and more numerous, right? There ends up just being a lot of them this side. Diffusion wants to take place. These want to spread out from this high area of concentration to a low area of concentration. And in the cell membrane, we give them an easy path to follow. There's an enzyme lodged here, that every time a hydrogen runs through this enzyme, kind of like a little a portal for it to go through, every time it goes through this enzyme, it turns. The enzyme turns. And when it does, it grabs an ADP and a phosphate, and forever and always, ADP and a phosphate will always make ATP. So every time something goes through, we make ATP. Those hydrogens will be, hydrogen ions will be recycled. Some of them will attach with the electrons to form an NADPH. Others will be just pumped back through, and this process goes on and on. And the name of that molecule that, or the enzyme that everything goes through to make ATP, it synthesizes ATP. Synthesize just means to make, so it makes ATP. It synthesizes ATP. What do you think the scientists called it? ATP synthase, because adding ASE on the end of something makes it an enzyme. So this is the process of the light-dependent reaction. Light comes in at two different places, photosystem 2 and photosystem 1. First time water is split, hydrogen catches the photon, its electron does. The proton goes off into the lumen. It goes down to a small ETC where it expends its energy. The electron hits photosystem 2 where it's recharged. Then it's from there it's attached to a proton, then attached to a NADP to form NADPH. And that transport molecule will then safely transport it over to the Calvin cycle. The rest of the hydrogen ions are simply run through a pipe that causes the pipe to spin. When it does, ADP and a phosphate is connected with another phosphate to form ATP, our energy molecule. So, and that also will go off into the stroma and be used in the Calvin cycle. So we've been able to account for everything so far in the light-dependent reaction. So the Calvin cycle is a little more complex. There's other things that are, a lot of stuff that's happening here. In order to capture carbon dioxide and bind it down and add hydrogens to it, we need a kind of a robust molecule to start with. And so the whole process starts with a molecule called RUBP. RUBP is a 5-carbon molecule, and we have to start out with 3 of them. So it is important in this regards to kind of keep along with the math that's going on, because we have to be able to account for all of our carbons. So we're going to start off with RUBP. It's a 5-carbon molecule. There's 3 of them, so we have a total of 15. The next thing we're going to do is we have an enzyme called Ribisco that's going to capture and bring in the CO2 from the air. It brings in 3 of those CO2s, so that's another 3 carbons. It's going to, Ribisco, the enzyme Ribisco is going to add this 3 CO2s to the 3 RUBPs. When it does, when you add carbon to something, you fix carbon to something, we call that carboxylation. And that's the name of this process. The end product that we get is 6 molecules of something called PGA. PGA has 3 carbons. So if you do our math, we started off with 15, right? Started off with 15 right here. Then we added 3 more. So 15 plus 3 is 18, but look how it's divided. It's in groups of, it's in 3 groups, 6 groups of 3, 6 times 3 being 18. Why PGA? PGA is a temporary storage molecule for carbon. That's pretty much it. We don't know if the plant actually needs all this carbon right now. It depends on things like if this is during the daytime, then this is going to be, yeah, you could probably use it right off the bat because you're probably running the cycle. But later in the afternoon when the light levels are down, or maybe cloud comes over, you're not going to be using all of the CO2 that you bring in, but we can store some for use later on. And the storage molecule for this is called PGA, and it is a 3-carbon molecule. Plants that use PGA are called C3 plants, and that's just kind of a stereotypical plant that we use with photosynthesis. It's just a 3-carbon C3 plant, meaning carbon 3 plant. And this is temporary storage. This first step, this first process is called carboxylation. Visco attaches carbon to RUBP to form PGA. Next, we have another step that we're going to go through. And in this step, we're going to take we need ATP to start off with, right? ATP is going to come in, it's going to get used up to drive the process. ATP will always break down into ADP and a phosphate, right? We start off with 6 ATPs, we end up with 6 ADPs. Next comes in is our NADPH, going to deliver those hydrogens. So there it goes. And as it does, it delivers those hydrogens. The hydrogens are going to be attached to the end product molecule. So when the NADPH drops off the hydrogen, it's back down to NADP. NADP would just simply go back to the light-dependent reaction, as well as does the ADP and the phosphate. So in all of this is where the hydrogen is brought in, and we use a little bit of energy to help bind it to a molecule. The end result is a molecule called G3P. G3P is also 6 carbons. And in this process, this is called a reduction, because we're simply reducing a PGA down and adding hydrogen to it to make it a little bit more ready to be turned into glucose. We're not quite there yet. Now notice that we produce 6 3GPs because we had 18 carbons when we had the PGAs, 6 PGAs, right? We didn't add any carbons, we just added hydrogen. So we're still at a total of 18 carbons. 3 of those are going to be in each of the G3Ps. Of the 6 carbons that we've made, or 6 molecules that we've made so far, 1 of them leaves out and is going to go towards glucose, right? The other 5 are going to be reused and modified back into our RUBP. But looking at the 3GPs, 3GPs are only 3 carbons. 1 of those per turn is going to be used towards glucose, but glucose needs 6 carbons. So it stands to reason that it would take 2 turns of this wheel to make 1 glucose molecule. So it would take 2 3GPs in order to make 1 glucose molecule. So that'd be 3 carbons for the first one, 3 carbons for the second one, and then add those together, and we end up with our 6 carbon glucose. So looking at the 5 of 6, the 5 of those 6, 5 of the G3Ps that go on. So we're not done with the cycle yet. We have to get back to the RUBPs. First thing we need in order to do this is we need some energy. So we're going to use a little bit of ATP. It comes in, ATP will always break down into ADP and a phosphate. When this happens, we simply regenerate the G3Ps back into RUBPs. Because let's see, we ended up with 3 carbon molecules. We end up with 3 5-carbon molecules. If you do the math, 3 times 5 is 15, 5 times 3 is 15. So we've accounted for all of the carbons in this cycle. So carboxylation, reduction, and regeneration. That's the 3 parts of the Calvin cycle. It takes 2 turns of this wheel to generate 1 molecule of glucose. And in this particular plant, PGA is the temporary storage for carbon. As we look into other plants like C4 plants and CAM plants, they don't use PGA. They use something else. So generally speaking, this is the Calvin cycle, right? And again, it produces glucose and it produces NADP and ADP and a phosphate. Those last 2 will be going back towards the light-dependent reaction. The glucose molecule will either be stored for long-term storage, turned into cellulose, or it could be broken back down through cell respiration and used for energy.